time, we'll quickly move to the next talk. We welcome Dr. Shridhar Narayanan. He is the Chairman and Chief Executive Officer, co-founder at the Foundation of Neglected Diseases Research. He has a strong drug discovery and development background with contributions to 18 clinical candidates across infection, diabetes, oncology, inflammation, pain, and CVD. He is the co-inventor of enmetazobactam, MVV 253, and leadership, and has provided research in TBA 7371. He's passionate about the high social impact diseases, especially infectious diseases and AMR, and that's what he'll be talking about today. Welcome, sir.
Very good afternoon. Thank you, Devanshi, for the kind introduction, and thanks to Abhijit, Abdul, and the WRCB team for inviting me to be part of this session. So I just wanted to start with, can I get a caller mic? Ask this question very frequently, so we work on rare diseases. And the answer to that is no, we work on neglected diseases. There's a very distinct difference between the two. Rare diseases are those which affect a very small population but have a very high commercial value. Yeah? So probably one of the drugs which was very much in the limelight between last year to this month. Source of disease, right? It's a rare disease. I work on neglected diseases, which are diseases which are usually affect a large population in the developing countries, and there is very little commercial value to them. Right? That's a very distinct difference. A lot of the neglected diseases are infectious. Development from my organization. For repurposed drugs is doing more than 50 research collaborations, right, across the globe. We have 10 grants from the government of India, thanks to them for supporting an organization like this, and several patents across disease indications. We also do some bit of service for our own survival. We are one of the only, I would say, not-for-profits which have a biosafety level 3 capability for doing both in vitro and in vivo work. work with some of the most dangerous organisms in the world, including SARS-CoV-2, which caused the pandemic. And we have completed more than 100 plus client projects with regulatory submissions being done both in India and across using our preclinical data. Right? Just based on my background in drug discovery, we also consult with a number of organizations with their own drug discovery and development. This is a small R&D center that I have in Bangalore. It's about 10,000 square feet of lab space with biosafety 2 and 3 capabilities and everything else that is required for doing preclinical research, including chemistry, cell biology, drug metabolism, pharmacokinetics, and chemistry. Right? We are fully compliant with all the government regulations, including the CPCSCA, DBT, RCGM, everything, and significant amount of infrastructure instrumentation, which is all funded through either grants or donations. So I just want to come to the main topic, which is how are we working in the One Health space? So we work on human health, on all these diseases. We have molecules in development. We work on environmental health through a device that we have developed which removes antibiotics from wastewater, which is one of the major sources of AMR. We also work with animal health, with zoonotic viruses which have pandemic potential, and also treatment of drug-resistant pathogens. This is my pipeline of molecules, which is there for the drug discovery stage. Our lead molecule is TBA-7371 for tuberculosis, and this is being developed in partnership with TB Alliance and the Gates Medical Research Institute. We're just completing a phase 2 trial in South Africa right now, and hopefully at some point, since this is a molecule invented in India, we want to bring it back to India and do clinical trials here in India. We have a bunch of repurposed molecules and formulations. These are molecules for broad spectrum antivirals with repurposing chloroquine for tuberculosis. We're working with a molecule called saracatinib, which is from AstraZeneca. This is being repurposed for TB as well. And obviously, the most interesting one for us is amphotericin B, which is an oral formulation of amphotericin B, which is not yet developed anywhere else in the world. Right? Plus, a series of molecules that we have which are in different stages of development. One collaboration that I wish to highlight is a collaboration between us and the National Center for Polar and Ocean Research in Goa, from where they brought back soil samples from Antarctica, and we've isolated some organism from the soil sample which produces a metabolite, and this metabolite works on a variety of microorganisms. Right? So we are developing that in conjunction with the National Center for Polar and Ocean Research. So this is our pipeline, like I said, 7371. It's a novel molecule, novel mechanism, a possibility of being a first in class when it gets to the market. So this is the molecule which came from Antarctica. We've done a variety of work on the Mycobacterium tuberculosis with this molecule. It's highly effective, where the MIC is between 0.25 to 1 microgram, including the MDR strain. So this is the data from the MDR strains, where you can see that across the panel, this is highly effective against the MTB. We've also looked at non-tuberculous mycobacteria, much more potent in the NTMs as well. It works against all the intracellular TB, the non-tuberculous mycobacteria, and also the biofilms, right, which is one of the most difficult stages of the mycobacterial growth to be treated. From the development angle, it has very little toxicity or no toxicity that we've observed, no cardiac liability and no CYP liability. And we know what the mechanism of action is. This is a study which was done by one of our collaborators, and this is a 6-month-long dosing in a model for TB. And we have seen that our molecule, which is this molecule labeled as NF1001 here, this one works all the way up to 6 months and actually is much better than any of the standard of care molecules. So we are looking at developing this as a molecule for the treatment of drug-resistant tuberculosis. We also have a pipeline of molecules for viral infections. So we are capable of doing any of the respiratory viruses, including H1N1, SARS-CoV-2, RSV, and we have a very broad spectrum molecule which seems to be working on all of these species with great potency. This is just some data from that. So this is a molecule which is from our stable, and this is a nice IC50 curve for this molecule also work on a variety of bacteria, fungi, and parasitic infections. So this is something that we've been doing in the last couple of years. We have one molecule for leishmania, which is being developed. We're doing some testing with CDRI Lucknow on this one. 20123, it's an anti-malarial compound, which is a first in class. I'll talk a little bit about that. So this molecule is an oral anti-malarial drug, which is a first in class HDAC inhibitor, which for the treatment of malaria. This works against all the strains of PF, Plasmodium falciparum, which are even resistant to the known drugs of malaria. And these molecules have very good drug metabolism, pharmacokinetic, and safety profile. And we've completed even part of the IND enabling studies with this molecule. We're looking at pushing this molecule into development both alone and in combination with other anti-malarial drugs. Coming to our other aspect that we work on, we are also developing a diagnostic to differentiate between bacterial and viral infections, which is going to be a point of care device using a blood-based biomarker. And we've actually done a 75 patient clinical trial with, in association with Amrita Institute in Kochi. And this is data from the study where we can see that this particular biomarker is increased significantly in a bacterial infection, but not so greatly in a viral infection. We are combining this biomarker with some of the other known biomarkers to develop a point of care kit for differentiating between bacterial viral infections, which will guide antibiotic therapy. So this as far as the human disease goes. In the animal disease, we are working on a variety of vaccines, therapeutics, monoclonal antibodies with companies both locally in India and abroad to develop these things. The last thing I wanted to touch on was a device that we have developed, which is for which we have filed a patent. This one basically removes antibiotics from wastewater, which could lead to antimicrobial resistance. And we have done this actually in the lab for 20 elutions, and you can see more than 90% of the antibiotics are removed. So this scale goes from 0 to I think less than 10%. And majority of the antibiotics, we have looked at 11 different classes of antibiotics and other drugs which are frequently prescribed in India, and it removes all of those from the wastewater to a significant extent. We've done a field trial in a hospital which uses these drugs, and we've shown that there is complete depletion of the antibiotics from the wastewater which comes from the hospital. And this after they have gone through the effluent treatment plants of that particular hospital as well. So we are trying to find a commercial partner for this who will take this forward and actually help us in deploying this. The entire device costs less than 1,000 rupees. So that's an indigenous invention which we want to take forward. In addition to this, as part of our mission, we also do a lot of community outreach. We work with school children across the board. We create awareness about the neglected diseases, especially localized neglected diseases like rabies, which are highly prevalent, snake bite, which is highly prevalent. We work on skill building workshops with the students. We invite students from different biotech schools, pharma schools to come and work with us. And we also organize industry visits. We provide support to girl child. If somebody wants to come and do training with us, we get them trained and also support their education and give them the time to go and attend college during or after the work. So that's something that we do as an organization. And we work with students across the board. If you have samples which you want to get tested in any of the infectious diseases, my organization will do it free of cost, right? So that is the support that we give from our organization back to academia. We work in collaboration across the board through small colleges, large colleges, IITs, the IISCs of the world. We're very happy to help anybody who wants to work in the infectious disease space. So if you have a project which you have in mind but you don't know where to go for a BSL 3 level work, come talk to us. We'll help you. This is my team. This is my co-founder, Dr. Shandil. And all of this work is done with a staff of 20 scientists, right? And a budget of less than 10 crores. Thank you so much for your kind attention.
request the
We'll just take one question from Amjad and that's it. Thank you.
Thank you for a very exciting talk and super exciting social services that you're doing. My question is more on when you talk about inhibitors targeting those bacteria or whatever. So like HDAC inhibitor repurposing, do you also take into account some of the very solid exclusion criteria when you do clinical trial? Let's say a patient who has HDAC mutated. So then, yeah, do you consider these things, and it can be more personalized? I just want to know your comment on that.
In the interest of time, the next talk by Professor Saurabh Ghosh, he has kindly agreed to let us move that after the panel discussion in the afternoon. So we'll request our next speaker after him, Mr.